So we're thrilled to have Dr. Kris Nichols with us today. Kris is a leader in the movement to regenerate soils for healthy crops, food, people, and the planet. A senior science advisor with us at Canadian Organic Growers, and the founder and principal scientist of Kris, Knowledge for Regeneration and Innovation in Soils, Systems Education and Consultation. Previously, Dr. Nichols was a chief scientist at Rodale Institute and a research soil microbiologist with the USDA. And throughout her career, Kris has given over 300 invited presentations to a wide variety of audiences throughout the world, authored and co-authored more than 25 peer-reviewed publications, and been cited or interviewed for more than 50 written publications, including articles and books. So today, we're lucky to have Kris here explore the why, how, and what of soil testing. So let's get into it. Over to you, Kris.
Thank you very much. And I am in Alberta, and we are having some storms coming through. So if there are any issues with the connection, please let me know, and I will try and address that as best as possible. You know, it's weather on the prairies, as many of you who are on this call know. So as David mentioned, apologize, David, as David mentioned, we're going to be talking about soil testing and discussing a little bit about the why to do some soil testing, some of the how, and then we're going to spend a little bit more time on discussing what to test for and what do these tests mean. And I'd like this to be fairly interactive. So I will have various points that I'll stop throughout this presentation for you to be able to ask some questions, and hopefully we can get some dialogue and discussion about this. So when we're thinking about why to test, there are a variety of reasons to go through and be doing the testing that you can have done on your soils, and a variety of different types of tests that you could utilize to address different issues that you might have going on. As David mentioned, through the Canadian Access Project, or CAP, we did some soil testing with many of you, and some of you are also involved in a couple of other programs that Canadian Organic Growers have been running for the last few years. The GEO program in Ontario, where there were soil samples that were collected as part of that program, and the RU program that is in Saskatchewan, where we're also collecting some soil samples. And really what we're trying to do through the CAP project, as well as through these other projects, is to take some soil samples and do some analysis that is a little bit different than what you would normally be doing. And part of this is in an effort to be able to capture the complexities that are involved in organic agriculture. In the past, a lot of soil testing has been very focused on the chemistry and what I oftentimes refer to as sort of in-out nutrient dynamics. So you're looking at your soil test to try and figure out what the nutrient status is so that you could determine what chemicals to be adding to that. And it's very defined within conventional agriculture, as opposed to looking at a lot of these complexities that are involved in organic and regenerative agriculture, where we're utilizing more diverse crop rotations. We're going to be utilizing green manure cropping, utilizing various types of biological amendments, utilizing compost applications, manure, a number of different types of complexities in these production practices that can make it difficult to be able to assess what's happening in your soil, when the tests are mostly designed, as I said, with looking at the soil chemistry and the in-out of nutrient dynamics. So throughout this presentation, we are going to be talking about that chemistry analysis as opposed to physical and biological analyses, and how to create and frame an entire picture to get a better understanding. Within the CAP project, where you're also utilizing this data to look at soil carbon dynamics and GHG emissions in order to be able to eventually look at carbon modeling and marketing as a potential leverage for producers to help to manage some of the risks that they may be confronting when they're trying to adopt new practices. So that can be one reason that you might be doing some soil testing. Another reason that you on your farm might be doing some soil testing is to be able to track what's happening with changes in management, to make comparisons between different fields or different areas within your field where it's under different management schemes, tracking that data over time and seeing what those changes are happening as you're putting those different management practices into place. And of course, increasing your knowledge. As I said, a lot of the traditional soil testing in the past has been very focused on chemistry. We're now transitioning away from that sole focus on chemistry to have a lot of the soil testing laboratories doing additional analyses for soil health that include physical and chemical and biological parameters. So we're trying to get a better example of what's happening in the soil as a whole. In addition, as we look, as I mentioned, this whole idea of these complexities within the system, where we've got all of these different types of practices that we can put into place. And we also can be doing some sampling within the CAP project that we're discussing, where we're going to be, as I mentioned, sort of leveraging the carbon markets, help to model carbon sequestration, and to mitigate various types of financial risks that you can have. In addition, the CAP data, as I mentioned, we're collecting that across Canada and looking at different types of agriculture and different management practices. So we will be presenting more of that data in the near future and sharing what it is that we have captured through this analysis. As David mentioned, this is a 2-year project that is coming to a close. So we are in the process of doing our writing of our final reports and collecting the rest of the data so that we might be able to present that to all of you. In addition, excuse me, in addition to the CAP project, I really encourage all of you, when it is that you're doing soil testing, to ask questions. Ask questions of yourself, of why it is that you're doing this testing, of what analyses should be done, what it is that you're looking for. How the analyses are conducted is something that you can ask the laboratories, and we'll talk about some of the importance of really addressing how some of these analyses are being performed. In addition, trying to get an understanding of what does the data really mean. And that's something we're going to be talking about this evening, but it is also something in which you can have a dialogue with the different laboratories and trying to get a better understanding for how this is looking as a whole and what the data means as a whole, as opposed to on an individual basis. So that's really, the foundation of why it is that you would go through and do soil testing. Just to give you an idea of how to go about doing some soil testing, and as part of the CAP project, we have put together a methodology for doing soil testing that we will share. But just roughly, soil sampling, soil sample collection can happen. There are certain private industry labs that will do soil sampling, both public AAFC and researchers with AAFC and universities may be doing some soil sampling. And you as an individual can also do soil sampling and collect your soil samples. And when we're looking at how it is that you go about soil sampling and creating a sampling design, there are a number of different ways in which we can design where it is that we're going to be taking the samples throughout the field. And so oftentimes how it is that we go and design where we're going to be taking samples depends a lot on the questions that you're trying to address, as well as what it is that your field looks like. If there are a lot of topographical changes, or if you want to be doing some zone sampling based on different types of management that you might be doing in a particular area, that's a way in which you might do the design a little bit differently than just going out there and laying out a grid that you can see here, an example of a grid pattern type of sampling, or that you would do sort of a bulk random sampling, which is up at the top. So you can really have different types of designs for the soil sampling, depending, again, on the questions that it is that you're trying to address. And CAP has a previous webinar that they've done as part of other projects that goes into a lot of details about the sampling design and the choices that are made for that. But I just wanted to kind of give you a little bit of an introduction to sampling design and what that might look like. So if you're ever wondering, if you're taking soil samples or someone else is taking soil samples and they say, well, go out and sample this way, why that's being done, that is also another opportunity for you to be asking questions. I really want to emphasize that point of asking questions, because that is a really critical component to what it is that's driving what we're trying to do, is answer these questions. And every individual has different questions that they may be trying to address. So it isn't a simple thing for us to say, just do it this way and just do these tests, because that doesn't necessarily address those questions that you may be having. So you really want to be able to kind of have that dialogue with the testing laboratory, as well as having a dialogue with whomever you might be working with on doing the soil sampling and analysis and interpretation. When it is that you're doing soil sampling, there's a variety of tools that you can utilize. Primarily, the main tools are listed here. Utilizing a soil sample probe or a shovel is typically how the samples are collected. You want to make sure that you're collecting the samples to a particular depth. So measuring how deep it is that you're collecting those soil samples. Utilizing various types of tools to make sure that you can easily collect the soil samples are listed here as well. Again, I don't want to go into a lot of details with how to collect the soil samples because that has been addressed elsewhere. When you collect the soil sample, it is important to note where you've collected that soil sample. So providing that information and history can be really important. And you can put the sample in a bag that you might be getting from the laboratory, or just putting it into a Ziploc bag. You want to store the sample in a cool, dark area until you ship it to the laboratory. And of course, following the laboratory recommendations or instructions on how to ship your sample and what it is that you're asking for as far as the tests to be run. That submission form and information and history can be very critical to the interpretation that is given, as well as for your own knowledge base to understand how the soil samples were collected, what has been happening in that field over time, where it is that you, what you might be doing in the future. All of those details can be really important to the laboratory to be able to do different types of interpretation. So I've gone over a couple of sections kind of quickly because, as I said, I want to spend the bulk of the time within this webinar talking about what to test for and how to interpret those tests. But I wanted to pause here before we start talking about what to test for to find out if you have any questions up to this point that we can look at addressing. So feel free to either raise your hand or to take your microphone off mute to ask questions.
Greg, you've unmuted. Do you have a question?
Okay, so if there aren't any questions, I'm going to move on to discussing what it is that you're going to want to be testing for. And when it is that you're looking at what you want to test for, you're really sort of an inspector of what's happening in the soil. And for me, as David mentioned, I'm a research scientist. And so oftentimes the questions that I have are very different than the questions that you have as a farmer or rancher. But it also is the case where the way that you look at your soil is very different than the way that I look at your soil. You know more about your farmscape than I will ever know. No matter how many tests that I run, you're the one who really understands what's happening there. And so as part of this inspection of what's happening and investigation of what's happening in your soil, there are a number of on-farm types of tests that you can do, looking at things like water infiltration rates, looking at soil structure, looking at aggregation or the slate testing, looking at basic decomposition processes through burying cotton cloths or tea leaves in your soil to see how the microbial community might be interacting with that, measuring compaction, just even the process of picking up a soil sample and rubbing it through your fingers and smelling it and looking at how that soil looks. That can be a way of interpreting what's happening on your farm. So those are a number of tests that you can do, the on-farm types of testing, and you can also do the laboratory testing. And again, we're going to be focusing more on the laboratory testing and those test results and what that information means and how to interpret those test results. So what you're really trying to do with the testing, again, is trying to address those why questions oftentimes is what's happening with the soil testing. Why is this particular area or this field performing this way? Why am I seeing the plants behaving in this particular fashion? Where are there some stress issues? What is it that I could learn from that soil sample? And as I mentioned, there are a number of chemical, physical, and biological tests that we can do. The chemical tests are tests that have been performed for a very long time on our soils. And for the most part, the way that the chemical evaluation of the soils has looked like in the past, and it still extends this way somewhat too, when we're looking at the chemistry, is we're looking at the chemistry in the soil for how it is going to impact these essential elements that the plant needs. So there are a number of other elements that are in the soil, but what we want to focus in on are these essential elements that the plants are needing. When we just take, however, a chemical perspective to this, we're not fully thinking about how the other elements that are in the soil and how the organisms that are in the soil and the chemistry and the biology and the structure, physical structure of the soil can impact the availability and the concentrations of these various essential elements. So if our perspective is just from the chemistry of these essential elements are what I'm looking for, and this is what I'm going to measure in the soil, we aren't getting a complete picture of what could be happening in the soil and how those essential elements may become available at different times throughout the growing season. And that's really where, when we look at an organic and or regenerative approach to agriculture, we really need to be focusing in on these types of concepts, because it isn't just, again, about the chemistry, but it's about the interaction and the nexus of everything that's happening. When we look at the chemistry, oftentimes we focus in on things like pH, because that can impact the availability of various types of nutrients. Some nutrients are more available at higher pH levels, and some nutrients are more available at low pH levels. This oftentimes really impacts the micronutrient availability in our soils. And sometimes that micronutrient availability, you can find issues with micronutrient toxicity. So with the micronutrients, the difference between deficiency and toxicity, that concentration can be very, very narrow, because these are micronutrients. They're only needed in very small concentrations. And so when we look at those micronutrients and we look at some of the test parameters, in some cases it may say that you have very high concentrations of various types of micronutrients. But if you also take a look at the pH, you can get a better understanding of if that fact that you have very high concentration could be a problem leading to toxicity. And so it's important to not just look again at the strict amounts that are given within the soil test, but also looking at the broader picture. How is the pH going to affect the dynamics of that availability? And is it going to mean that I'm going to have a deficiency issue, or is it going to mean that I'm going to have a toxicity issue? And so again, looking not just at individual numbers within the soil tests are going to be really important. And as we look at some of examples of some soil tests, we're going to go through and talk about that a lot more on what might be happening. So I've got here the whole idea of macro and micronutrients, and then the statement saying available, extractable / potentially available, and total. And what has happened in the past, again, because the way that we looked at the chemistry in the soil was not really that the soil was a media that had biology and chemistry and physical activities going on all at the same time. It was looked upon as a media where we would extract nutrients and we would add nutrients. So it was very that what I have said before is kind of the in-out type of chemistry. If you extract or harvest a crop, you're taking a bunch of nutrients off of the soil, out of the soil. And so don't you have to put those nutrients back in? And that may or may not be the case. It oftentimes depends on what the total concentration is and what the extractable or potentially available concentration of nutrients might be. So as we've looked at some of the tests that have been conducted in the past for measuring the concentrations of various types of macro and micronutrients, that is focusing on more on the available concentrations or the concentrations that are extractable with strong acids, but not looking at the total or what the microbial population and the biology interacting with the chemistry and physics may be able to make potentially available. So there are a number of tests that are now being conducted to try and get more information about the total and the potentially available nutrients that you might have in the soil. So the standard analysis, again, going back to this, the standard analysis, if you just do standard soil testing, will give you most, if not all, of these essential elements, depending on the test that you choose. And that will be focused primarily on the concentrations of those macro and micronutrients that are available. And it's also key to understand that that is what the nutrients that are available at the point in time in which those soil samples were taken. And as we all know, temperature and water concentrations, soil water concentrations, as well as biological and physical parameters can change throughout the growing season and throughout the year. And that's going to result in us having different concentrations of available macro and micronutrients at different times. So when we're looking at these soil test results, it is a shot, just a picture at one point in time. It doesn't always tell you about everything that might be going on or what might be available when it is that you go through and are managing your crop plants. So we really need to take this, I say, take it with a grain of salt in this understanding of what the chemistry is telling us, the chemical tests are telling us as far as our nutrient concentrations, and if we are really mining nutrients out of our soil, or if it is that we have not a large concentration of nutrients, but they just aren't all in that available form. And so how is it that we can go through and make these nutrients more available? One of the other tests that is becoming more popular is looking at soil organic matter. And organic matter, we'll discuss this a little bit more in detail, but when we're looking at organic matter, we don't just want to look at organic matter itself, because that's a very complex group of molecules. One of the other things that we're looking at, and with the CAT project, we're looking at total carbon, organic carbon, and inorganic carbon to understand more about that carbon dynamics and how organic matter is interacting with the biology, as well as the potential to build up that organic matter in the soil. So we really, again, with the chemistry, we've got these key tests that it is that we look at, but we don't just want to look at the chemistry. And so we've changed a lot of the testing that we've done to now include additional physical and to include, starting to include biological parameters in our testing. With the physical parameters, as I said, you can do things like water infiltration. We can look at bulk density. That is basically in a column of soil, how much is filled with solids versus how much is filled with pore space. And that's going to give us an idea of that pore space, that open space that could benefit water infiltration rates, increase water infiltration, can also help with water retention and water holding capacity, can help with aeration and gas exchange between the microorganisms and the plant roots and the atmosphere above the soil, and looking at soil texture, things like the concentrations of sand, silt, and clay that can impact the levels of compaction, can impact the ability for the plant roots to grow, that porosity that you would have, all of those things that can be really important as soil health indicators that are associated with these various types of soil physical, chemical, and biological attributes that you can have. Biological testing that we can do, we can look at what are some of the particular organisms that are in the soil, utilizing a microscope to examine the organisms themselves directly. We can also look at phospholipid fatty acid concentrations, or PLFA concentrations, that we can then use to group organisms into various types of potential functional groups that could allow us to be able to get an understanding for how the biology is interacting, again, with the chemistry and the physics within our soil. We can do DNA analysis, which can give us an idea of who's there, but we can also do analyses looking at activity and viability of the various types of organisms that are in the soil, the enzymes that they're producing, and how that impacts functionality, as well as metabolomics, which is associated with how they're functioning in the soil, what it is that they're doing. Oftentimes, again, when we're looking at soil health and looking at soil testing to get an understanding of soil health, it has a lot to do with the functionality of the soil. What is it that we want to get from our soils? And that becomes part of the question of why we test, is what are the functions that we're looking for? If I have issues with compaction, are those issues with compaction because of my soil texture, because of my organic matter concentrations, because of the amount of porosity that I might have in the soil? All of these play different roles and interact. And again, they're not values and numbers that we can examine individually, but it's actually this more complex type of interaction that we're looking at. Here's a listing of a bunch of other tests that can be done in addition to the standard or additional laboratory tests that are out there with commercial labs. So these are tests that a number of different types of researchers might be doing, different industry partners might be doing, different types of organizations and associations might be looking at conducting on your fields. In addition, if you look towards the bottom of this list, we're going to talk, it lists some of the plant parameters that could also be looked at to get an understanding of what's happening within the soils themselves and how the soils are interacting with the plants and the plant productivity and plant growth. So I'm going to pause here,
Kris,
and
sorry,
pause for some questions. Yep.
Okay, yeah, great. I just wanted to say it's 36 after 7, after the hour.
Okay. So I wanted to pause to see if there are any questions.
I have a question.
Yes.
What is the difference between organic matter and total organic carbon?
So organic matter, and that's a very good question. So organic matter is a large suite of a number of different compounds, from sugars to proteins to carbohydrates, to the bodies of microorganisms, to root debris and plant leaf debris. And it's all of those different types of compounds. And when they refer to it as organic matter from a chemistry point of view has carbon, hydrogen, oxygen, and some nitrogen. But when we're looking at total carbon, that is the total amount of carbon that's in your soil. And that carbon can be carbon that's in that large suite of different compounds that are part of the soil organic matter. But you also can have carbon that may be part of the inorganic fraction, which is things like calcium carbonates or other types of carbonates. Calcium carbonates are a lot of the lime that you might have and limestone that you could have in your soils. And total organic carbon is the carbon that is associated with the organic matter. But when we're measuring total organic carbon, we're just measuring the amount of carbon that's in that organic matter. So I said that organic matter is carbon, hydrogen, oxygen, and some nitrogen. And when we're looking just at total organic carbon, we're looking just at that carbon amount that we have in the organic matter.
Thank you.
Yes. So when we talk about what do soil tests mean and what it is that we're looking at, I hope that you've kind of over the last half hour or so gotten the perspective that this is not just one thing, but it's everything interacting in a natural system. And I emphasize the biology and the chemistry and the physics, but the geology that is part of that sand, silt, and clay concentration, it's part of the other micronutrients that may be within the soil. All of that is part of that natural system. And so when it is that we're looking at the soil tests and their meaning, we need to look at it from the whole system approach as opposed to individual numbers. That whole picture where we're looking at soil functions and interactions. And when we do this, we're going to get a better understanding for the function and being able to address the questions that it is that we're looking for. And again, these different tests, as you can see from this diagram, as well as the previous diagram, these different tests and these different features to the soil are all interacting and overlapping. The chemistry doesn't just happen on its own. Physics and biology also impact the chemistry. So we've got all of this complexity that's happening together. As I mentioned before, when it is that we're looking at the standard tests that are there, oftentimes the values that we're given from just the chemistry point of view are values for micro and macronutrients that come from extractions, chemical extractions that are assessing that whole idea of what could be available to the plant, not what the biology and the chemistry interaction could make available, but what could be available to the plant at that one point in time. So again, it doesn't necessarily reflect the total mineral concentration in the soil. And that's why it's important for us to look at all of these interactions. Many soil testing laboratories won't measure nitrogen, or if they do, it will be a separate test in which you have to request, oftentimes to get nitrogen measurements as nitrate nitrogen or ammonia nitrogen that may be happening. And that's because nitrogen is fluctuating within the soil due to these interactions of physics, chemistry, and biology, so that it's hard to be able to assess and quantify nitrogen directly. In addition to that, when we're looking at nitrogen, as David mentioned, one of the tests that we've added to our analysis within the CAP project is looking at carbon and nitrogen ratios. And within that, we're trying to take a measurement of the total nitrogen that's in the soil. So again, oftentimes with the chemistry tests, if they do look at nitrogen, they want to look at nitrate nitrogen or ammonia nitrogen, because those are the forms of nitrogen that have typically been looked at as being the forms of nitrogen that are going to be available to the plants, readily available to the plants. More recent research has shown that plants also have the capability to take up various forms of organic nitrogen. So nitrate nitrogen and ammonia nitrogen are what's referred to as inorganic nitrogen. When we're talking about this, this is different from referring to things as an agricultural practice that is organic and an agricultural practice that's not organic. What we're talking about there in reference to nitrogen or other elements like carbon, when we talk about organic carbon versus inorganic carbon, that doesn't mean that one is formed by organic agriculture and one is formed by non-organic agriculture. What we're referring to is the chemistry actually of that carbon, what it looks like on a molecular level. And so in organic carbon or in organic nitrogen sources, that nitrogen source or that carbon is a combination of carbon, hydrogen, and oxygen, or organic matter, that chemistry that's there. And if it's organic nitrogen, it will be carbon, hydrogen, and oxygen and nitrogen. Inorganic carbon, nitrate, and ammonia, nitrate is NO3, so it has nitrogen and oxygen, but it doesn't have the carbon associated with that and the hydrogen associated with that. When you look at ammonia, that is NH4, and in that case, you've got nitrogen and hydrogen, but you don't have, again, carbon and oxygen associated with it. So for something to be considered to be organic nitrogen, that nitrogen has to have carbon, hydrogen, and oxygen associated with it. And usually that nitrogen is in the form of proteins or amino acids. And the reason I bring this up is that, again, from the standard soil testing that's been done in the past, we focused on the inorganic forms of nitrogen that are typically fluctuating very rapidly through the soil. So we don't oftentimes measure those as a standard analysis, but an add-on analysis. And the reason they're fluctuating very rapidly in the soil is that naturally those compounds aren't very stable in those inorganic forms. So when we're looking at organic forms of nitrogen that come from proteins and from amino acids, those forms of nitrogen are more stable and they don't fluctuate quite as rapidly. So within the CAT project, we wanted to get an understanding of the amount of carbon and the amount of nitrogen that we would have, because we know that the biological activity is actually assisting in helping to make sure that that organic nitrogen is going to be able to be available and utilized by the plant. And the plant, as I've said, is now more capable. We have a better understanding from a research perspective that the plants are capable of not just absorbing inorganic nitrogen, but also absorbing organic nitrogen. Again, when we're looking at the soil testing, a lot of what we're looking for in the soil testing and a lot of the answers that we're looking for is tracking over time or getting a metric that is repeatable, that's something that we can continuously get measured and be able to do these various types of comparisons. But again, we need to be looking at everything as a whole. So when we're looking, and to the question that was sort of discussed before, again, what does organic matter versus carbon mean? And when we're looking again at organic matter, it is carbon, hydrogen, and oxygen with a minor amount of nitrogen. And so it isn't just one molecule itself. When we look at this, there is all of the organic components that are parts of the bodies and that are the biomolecules that are produced by the organisms that are in the soil that are part of the organic matter. It is also more decomposed types of humic materials. And it is that residue that may be coming in on an annual basis. And so when you've got all of these different compounds of all different structures, we call that all organic matter, but it isn't just one thing. And so when we're trying to make some measurements about what's happening with carbon and carbon sequestration, what it is that we're measuring is looking at the carbon, looking either at the total organic carbon or looking at the total carbon. And roughly the way that this is measured and equated to soil organic matter, because again, organic matter is a very complex, dynamic, different types of compounds that are there. So what they figure, a rough estimate of the amount of carbon that may be in organic matter is about 58%. And I have here, it says 58% of LOI. What does that mean? LOI is a type of soil testing. When they're measuring organic matter, it's called loss on ignition. And so what they'll do is they take a soil sample and they weigh it, and then they put it into a muffle furnace and heat it up to greater than 800 degrees and burn off the carbon that's there, burn off the organic matter that is going to be there. And when they do that, then they take the sample out of the furnace and weigh it again. And the difference between the weight before it went into the furnace and the weight after it went into the furnace is called the loss on ignition. It's the weight of, it's an estimate of the weight of soil organic matter. So in most soil test results, when you get an organic matter or soil organic matter value, it is that number that's based on this test of a loss on ignition. If we're going to be doing a measurement and trying to get a better measurement of the carbon that's in our soil, what we want to do is what's called dry combustion. And with that, again, we heat up the soil to a very high temperature, but then, and when that happens, that's going to convert the organic matter into a gas. That actually converts the organic matter into CO2 because we're burning it off, and we directly measure the CO2. And so when we do that, that measurement is a measurement of carbon as opposed to an estimate of the carbon that's based in the soil organic matter, and that we say is roughly about 58%. We can then take that carbon value and convert that to soil organic matter by multiplying it by 1.72. And so when you're getting soil testing done, one of the important things to ask is, how is it that they're going about measuring organic matter? And for the most part, for standard soil testing, getting measurements of organic matter based on loss of ignition can be just fine. But if we're doing things on looking at things like carbon sequestration and modeling the amount of carbon that we may have in our soil for carbon markets, that is when we want to get the dry combustion measurement that is a direct measurement of the carbon. And we're not caring as much about measuring the amount of organic matter that we have in our soils. So again, just wanted to give you another perspective on this complexity of what's happening and all of these different interactions that are occurring within the soil itself. And that organic matter is kind of at the nexus of the chemistry, biology, and physics. So it's one of the big drivers for what's happening in the soil. And when you have these inputs of carbon into the soil that can happen through various types of practices that you may have, utilizing cover crops, green manure, integrating crop residue, animal manure, animal grazing, perennials, diverse crop rotation, all of that can be different ways of building up organic matter in our soil that then affects the physical, chemical, and biological properties that you're going to have in the soil. So as we look, we're going to go through various types of soil tests that you might see, a soil test report that you might get from a few different laboratories. So we're going to start with A&L. And so this is an example of what A&L would send you as far as the analysis that they're performing on your soil. And so one of the first things to look at is to take a look at the organic matter that you have in your soil. And then you can start looking at, the next thing I would take a look at is looking at the pH that you have. So your organic matter in this particular sample, they have organic matter that ranges from 1.5, low organic matter, to a high of 4. And they have pH that is going to be ranging from 4.6 to 5.3. Neutral pH is a pH of 7. Most of the activity and most of the nutrients are going to be more available when you have pH levels between 5.8 and 6.7. So you've got a pH range in there where the greatest concentration of nutrients can be available. And then outside of that pH range, it doesn't mean that those nutrients aren't available. It's just that they're less available than if you were at that ideal pH range. So we would like our plants to be in soil that would have that ideal pH range. But obviously, in many of our soils, we can't get that ideal pH range. And when we're looking then, as I said, looking at the micronutrients that you might have in the soil, that is going to, give us an indication of the toxicity or the deficiency of various types of micronutrients. Now, in this case, there's very high levels of iron, high levels in several of the soil, or very low levels, of manganese, very low levels of zinc, and very low levels of copper and boron. So with some of the micronutrients, when you see those values for iron, we potentially could have a little bit of an issue with iron, but we're not probably going to have too much of an issue of iron toxicity because our pH levels, except for in the soils where the pH levels are below 5, you may run into a few concerns with the very high iron levels. The pH level that you have in the soil is actually going to assist in being able to get the copper and manganese and zinc that are in very low concentrations. Most of them are going to be available. And so that can be it can be beneficial at that pH level. So even though you've got very low levels, most of that is readily available to the plant. And so we need to be looking again at things as a whole. Organic matter is the ultimate buffer. And what I mean by that is at a wide variety of pH levels and a wide variety of concentrations of nutrients, if you have high organic matter levels, your availability of those nutrients is going to be buffered by the organic matter, and that's going to keep most of those nutrients, even at very high or very low pH levels, or very high or very low concentrations. It's going to keep those nutrients available in an adequate amounts that is going to buffer the needs that the plant has and buffer the issues that come from pH or concentration of the nutrients. So by being a buffer, it allows for those nutrients to be available at a wide spectrum of pH levels and a wide amount of concentrations. This may be another sort of viewpoint of graphical analysis report that you might get from A&L. Again, looking at those levels, but you don't want to just look at the size of the bars themselves, but look at not just the individual numbers, but how all of these different interactions may be happening within that system. This is A&L will do some biological analysis, so you can get some nematode analyses done by request. Again, that's not part of their standard packages, but it is something addition that you can add on if you've got issues, and you can add additional types of testing that you might be looking for. If you've got some issues that you might be looking for as far as salinity issues, you can add additional testing specific for salinity analysis. You do get a lot of information from this report, from the standardized report, on things like cation saturation and base saturation that you would have here. And that can give you a lot of information about salinity issues that you might have. We've got a pretty high concentration of calcium in these soils. And so you may need to be looking a little bit. Calcium and magnesium do have a tendency to play off of each other. So if you've got a very high concentration of one and a very low concentration of another, you can run into some issues with soil tightness, which can result in additional compaction issues. But from this analysis, the values of magnesium, the percent saturation of magnesium and the percent saturation of calcium are pretty close. So I don't think that you're going to run into a lot of issues there that you would need to look at resolving. You can also get analysis. It's going to include the soil physical characteristics or the soil texture. So we've got a wide range of soils that you've got here: a clay loam, a sandy clay loam, you've got a silty clay, and clay soils. What this means is that the minerals, the mineral rock has been broken down into various concentrations of sand, silt, and clay. And if you have a lot of clay in your soils, oftentimes you run into issues with compaction, because the clay minerals are going to layer tightly on top of each other in their structure, and there's not a lot of open space. They're very small particles. So as they layer on top of each other and interact, there's not a lot of space in between there for porosity and the open space. So you end up getting a lot of compaction. When you have very sandy soils, those sand grains are going to be very large, and you may have a greater amount of porosity in your soil, which could be good for root growth and for infiltration rates. But with very large pores, you may not be able to retain a lot of water in between rainfall periods. So ideally, the loam is basically, when something is a loam, it is a soil that is pretty much equally concentrated in sand, silt, and clay. So it's got a close to even distribution. Now, when you've got a clay loam, that means you've got a little bit more clay than sand or silt, but you still have a pretty good soil as opposed to having something that is a clay or a silty clay where you've got a lot of fine particles. One of the things that can help with soil texture issues is, again, as we start to increase organic matter, because organic matter not just buffers pH and nutrient availability, but it also can help to buffer texture. So it can add porosity to a soil that may have a high clay content or a high silt content. And it also can help to shrink some of the large pores that may exist in a very sandy soil and allow for the water that are in that pores to be able to be retained for a longer period of time. So again, organic matter is sort of the big thing that we're looking for here to solve a lot of our issues. Oftentimes, with the standard analysis that you also get soil fertility guidelines. Again, I would look at these in context and not as absolutes, because they are measuring the availability of those nutrients at that particular time. That doesn't mean that that's the amount of nutrients that would be available when it is that you go through and plant, or at different times throughout the growing season. In addition, if you're using regenerative and or organic practices, that is going to be changing that fertility and that potentially available fertility. That means that you may not need to follow these fertility guidelines. These were designed for conventional systems. So you need to think about what this means within your whole entire system. I'm actually going to come back to this. So I'm going to skip over this for right now. Couple of the other tests that I wanted to introduce you to, the Kris Soil Health Lab in Alberta, in Oyen, Alberta, is doing a combination of chemical, physical, and biological analyses, where they're breaking down various types of indicators and then coming up with a soil health score. And again, what we need to be looking at here is it's great with these soil health tests, because the way that this reporting structure has been designed is that we've got colors, and your eyes will naturally gravitate to various types of colors and associate that with things being good or bad. So wherever there's red, that's going to be something that you're going to associate with something being bad, or the values not being optimum. And when you have colors that are blue and green, that means that those values are more favorable. Where you have colors that are yellow and orange, those are moderate, they're in between. So our eyes will naturally go and look at this and look at those colors and associate this with good or bad types of things that you might be looking for. But again, we need to look at this as a whole. So the soil health score does take into account a number of different parameters when they're trying to look at this and try and look at this as a whole. But we need to continue to be able to do that, because part of that holistic interpretation, this test was designed, even though it's currently performed in a laboratory in Oyen, Alberta, it was designed at Cornell University in the United States in soils that aren't the same types of soils that we might be seeing in different areas throughout Canada. And although this test has been applied to a number of different types of soils, when they created their numeric system for evaluating the value and determining what's good and what's moderate and what's bad, a lot of that was done in more conventionally designed, more upper northeast United States types of soils, which are not necessarily going to be the same types of soils that you might be seeing across Canada. So when, again, your eyes go to look at the color, but the colors may not be as much of a value depending on where it is that you are. And how you can change some of that perspective and put that into account is looking at things like organic matter levels. So the organic matter levels in this particular soil are very, very high. Looking again at pH, the pH level is a high pH level. So where you may have some issues, where again it says things like excessive iron, that is coded in red. But with the pH value that you have here, because this pH value is above 6, and with the organic matter values that you have that provide for a buffering capacity here for nutrient availability, the excessive iron and excessive magnesium and potassium values are understandable, but they're not going to be causing you an issue that in a more acidic soil with a lower organic matter concentration, that would be more of an issue. So again, in looking at these tests, what they've done here is they've created a color code that is based on the individual number, but it doesn't take into account the full picture of what's actually happening in your soils. So when you look at organic matter and pH and your nutrient concentrations, your soil structure, you've got good aggregate stability, you've got a good bulk density number, you've got some compaction issues that exist here, but you've got a lot of good things to be building on from a physical health standpoint and from a biological health standpoint, which makes some of the chemistry issues that you see here and some of the compaction issues that you see here a little bit less of an issue. And so again, don't just let your eyes go to the color, but think about the tests as a whole. And that's really what I want to emphasize as we go through and look at these various types of tests. So these are the same tests done on some different soil samples. So they have different soil health score ratings. Should have different soil health score ratings. This is a 45. Sorry. So we've got all of these different types of soil health score ratings that you have, a 50 and 59 and a 45. And comparing what's happening in those different soils when it comes to that biological and physical parameters as compared to the chemistry parameters, and not letting one chemistry or biology or physics override the complexity that exists there. One last series of soil tests that I wanted to go through is through in the US, Ward Laboratories or Regen Ag Laboratories in Nebraska. Some of you may have sent soil samples for this type of testing, where we've got what's called the Haney soil test. And what the Haney soil test is trying to do is it is trying to capture what might be happening on a biological level for changing the potential availability of various types of nutrients. And so what the Haney test is looking at is it looks at the organic matter levels, it looks at the respiration and the biological activity, and it looks at the extractable fractions of various types of nutrients that can either be extracted in water or could be extracted in this H3A solution, which contains a lot of different types of organic acids that would be produced by the plant roots, as well as produced by the microorganisms, to help to make various nutrients more potentially available. So when we talked about the standard tests, they're looking at what's available to the plants at that time, but not on a chemistry standpoint, but not what biochemistry, enzymes, organic acids, what biochemistry could potentially make available. And so that's what the Haney test is trying to do, is assess what might be available over time. And then they do give a soil health score. And they also are going to give recommendations as far as cover crops that could help to make these nutrients more potentially available. So we've got the standard Haney test that you've got there, that information that'll be there. And then what they do is based on what you let them know as far as the different types of crops that you're going to be putting into the system, it's going to let you know what the nutrient recommendations are based on that potential availability of nutrients, as opposed to the A&L test that looked at what the availability of nutrients was at that particular time. So this Haney test has been something that has been utilized a lot more recently as organic and regenerative organic agriculture and regenerative agriculture have increased in usage. When you're utilizing more of these biologically based practices, that's going to change the amount of potential availability. And it's especially important for things like phosphorus. Phosphorus has a very low availability rating, even though the total amount of phosphorus in your soil may be very high. And that's because phosphorus gets readily bound up at both high pH and at low pH. So there's not really much of a pH window in which phosphorus is readily available. So oftentimes phosphorus is looked at being limited in our systems, but the biology produces various types of enzymes and organic acids that can help to make that phosphorus available, even if the pH isn't quite right for that availability. And that's one of the things that the Haney test will help you to get an idea of if that is available or not available. In addition, Ward Laboratories and other laboratories are doing this phospholipid fatty acid analysis of microbial communities. And what this does is it's a very broad stroke grouping of microorganisms. You have bacteria, gram-positive and gram-negative bacteria. You've got some key bacteria that it'll pull out, actinomycetes and rhizobia involved in nitrogen fixation, and actinomycetes that can also help with nitrogen availability. And then you've got arbuscular mycorrhizal fungi that can help with nutrient availability, and saprophytic fungi that are actually the decomposers. And so it'll break down the total fungi into these different categories. You've got protozoa, and then you've got here this large amount of biomass that is called undifferentiated. And this is stuff that we don't know. We either don't know what their fatty acid structure should look like, so it's difficult for us to be able to quantify them and group them, or they haven't really been identified. Some of these microorganisms haven't been identified at all yet. So there's a lot that we're still learning on a microbiological level to get an understanding of what might be happening in your soils. So just like with the chemistry tests, these biology tests are snapshots. They're windows in time and they're windows in time with the knowledge that we have on what's happening within those systems. There's some additional information and interpretation on what these different types of ratios of fungi to bacteria, predator and prey ratios, gram positive to gram negative, saturated and unsaturated, all of these things might mean as far as what it is that we have measured within these different organisms that we're taking a look at. But again, I always say with any soil test that you're doing, there are caveats, and you need to understand that they aren't showing you absolute values, but they're showing what it is that we can measure and that picture in time, as opposed to the broader range of complexities that are going to be existing. So I know we've kind of gone over time, but I want to know if you have any questions, or if you do want to spend a little bit more time. I wanted to go back to this particular set of A&L tests that we've done as part of the CAP project. So before I do that, I will open it up for questions.
I have questions, but they relate to a few of the things in the A&L soil test report.
Okay.
So you might be answering them. Like, as an example, what is ENR stand for?
What is ENR?
ENR, yeah.
Yeah. So that is, go back to this. The ENR, this is a conversion of the organic matter. Instead of it being as a percentage, it's a conversion to the pounds per acre. So it's just giving you that in a different value.
And then I also wonder why is the K to mag ratio important for them to? Why would I want that, I guess?
So in different soils, so you're talking about potassium to magnesium as opposed to calcium to magnesium?
Well, like
percentage?
most of the soil tests I get have the percent base saturation ratios, but I haven't had one before that includes the potassium to magnesium ratio, and so I was just curious about that one.
Yeah. So when we're looking at, and in many of our soils in Canada, it's not going to be much of an issue. We're going to be looking more at the calcium and magnesium types of dynamics that are going on. When we're looking at the potassium and magnesium types of issues, that is going to be in some soils that, again, are not necessarily going to be in Canada, but it's going to be an issue in some of our soils in, I do some work down in the desert Southwest, and so you're going to run into more of issues with potassium and sodium there and how that might be interacting with magnesium. So when we're looking at this base saturation, all of those cations are going to be interacting with each other. And what I mean by that, I guess they're not directly interacting with each other, but they're always competing for binding sites on organic matter, or competing for binding sites with clay minerals and other minerals that are in the soil. And so when you've got that level of competition between these different cations for those binding sites, the percentage of those cations can make a difference on which ones are going to be bound to more of the binding sites, so their total concentration, and the things like potassium and calcium and as opposed to magnesium, the size of those atoms, looking on an atomic level, makes a difference on how compacted or tightly bound things are. So if you've got a large atom like calcium or potassium that you've got there, that can help to open up soil structure because they're larger. And so it's the same as kind of having sand grains as opposed to clay minerals. So magnesium is kind of like the clay minerals that are in there. And those layers can be very tightly bound to each other. But when you've got calcium or potassium, you can have a much more open structure. And potassium is larger than calcium, and sodium is also larger than calcium and magnesium. So again, the more open, the larger the atom is, the more pore space you can have or open space you can have around those atoms. And that can affect the amount of compaction that you have.
Okay, thank you. And quickly, you said you referred to something as having some farm soil as having a good bulk density number. And
Yes.
so I'm wondering what is a good bulk density number?
Okay, so we're going to go here and look at bulk density. So with bulk density, what you want for bulk density is you want values that are going to be, in this case, they're looking at kilograms per meter cubed. Oftentimes it's also presented as grams per centimeter cubed. And so when it's kilograms per meter cubed, you'd want values that would be less than 1,500. If it's in grams per centimeter cubed, you want values that are less than 1.5. The closer you are to 1, the less dense your soils are. And so that means that you have a greater level of porosity. And that porosity, that open space, is really good for root growth, as well as for aeration and for water infiltration and water holding capacity. So the bulk density, again, is looking in a volume of soil, in a soil PED, how much of that soil PED or that volume is filled with solids. If you have something that is, if you have bulk density values that are 2,000 or 2.0, again, depending on if you're measuring it in grams or kilograms, that is starting to get to be very dense soils. And solids, a pure solid, so if the soil had no porosity and was a pure solid, you'd have values that would be about 2,600. So again, you're looking for values that are going to be close to 1, but definitely below 1.5 or 1,500 and close to 1,000.
Thank you.
Yep.
There's a couple questions in the chat. Kris, maybe you'd be able to cover them quickly. First one is Ezra is asking, are there ideal benchmarks for each element that they can compare their soil test results against?
Unfortunately, that's a yes and no answer. The yes to this is that for the most part, that's, when you do get soil test results and they give you values like, they discuss things like low, high, those types of things. That language there, low, high, moderate, very low, very high, is based on ideal values. And again, those ideal values are more based on conventional agriculture than they are based on regenerative agriculture. When it comes to things like the Haney test or some of the biological testing that's being done as opposed to the chemistry testing, those ideal values are a little bit better as far as the looking more at organic and regenerative types of agriculture. But the biggest issue with them as being ideal values is there's not enough data for us to know what those ideal values should be. And so there's a lot of assumptions that are made based on those ideal values. So again, with the soil test results, with the reports that you receive, you'll see things that'll say HML, indicating high, low, moderate. And you also, within things like the Haney test, you could see values where they will say, part of the reporting will be what the ideal should be. So with the fatty acid testing and with the Haney testing, it'll give you information in the reports as to what the ideal value should be. But again, you need to take that with a grain of salt, because we don't have enough data to really know for sure what those ideal values should be, and enough data across a large enough ecoregion. There have been some initiatives on both the Canadian as well as the US side to actually do large scale testing across a large number of ecoregions. For the Haney test, that was done on the US side, where they were trying to look, the Soil Health Institute was trying to look at I think 5,000 samples or so across the US to try and get that information. If you talk to a number of statisticians, though, a lot of times they'll say, well, you need more like 25 to 30,000 samples in order to be able to really get good data. And that's looking, just within the US as opposed to globally. So it is a thing that we're continuing to work on and trying to get a better understanding of. I wish, so that's my yes and no. I wish it was better.
Yeah.
Is it fair to say that the ratio
Thanks,
is more important?
so it is 8:30. We did this. Oh.
Yep.
You want to just let
so
David finish what he was saying?
Let's go. Yeah, go. No, it's fine. You go ahead, Kris, and I'll loop back.
Yeah. Yeah. I was just going to say that David, I know you had a question about the ratio. So if you want to finish that question.
Was it fair to say that the ratio is more important than the actual, whether it's high or low? It's how the ratio of the components together, I guess, especially on the micronutrients.
Yes, that is definitely, again, there are, as I said, a couple of different things. The ratios can be really important, as well as, again, the interactions with pH levels and organic matter. So the individual numbers, that's why I kept saying too, the letters that are there, or the colors that are in the Kris soil health test, anything that you have that's telling that, don't just pay attention to what the letters say or the colors say, but look at everything as a whole, those ratios, how things are balancing out between each other, and what that means from a pH and an organic matter perspective.
That's great. Yeah, we're dealing with complex systems, and there's no way we can cover it all, especially in an hour and a half. So it's 8:31. We did want to wrap it up around 8:30. There was one question in the chat though that was there for a while. So Kris, I was wondering if you can maybe spend about maybe in a minute or two, if you could explain the significance of cation exchange capacity.
Yes.
Or why should we pay attention to that on our soil test report?
So the cation exchange capacity is, again, looking at those cations that are involved in the base saturation, and how exchangeable they are. So what they're bound to, can they get bumped off? And what cation exchange capacity can give us an idea of is where it is that we can get nutrients to become more available. So as you look at those various cations that you have, if they are exchangeable, oftentimes that means that you've got that exchange, that when the cations are getting exchanged on a binding site, you're going to get anions or oftentimes nutrients like nitrates, sulfates, and phosphates that are going to be able to be released or become more available. So you don't want to have a lot of, if you have a very low cation exchange capacity, it means that those nutrients, nitrates, phosphates, and sulfates, are not going to be as available because you can't get the cations to get bumped off and make those nutrients where they're bound to more available. Again, it's more based on chemistry than it is on what the biology can do. And as you build up organic matter has both cationic binding sites and anionic binding sites. And so that's where, when you get either cations or anions bound to organic matter, they're much more exchangeable because organic matter binds them in a shared bond. Whereas if you're looking at cation, anion bonds that you have, that exchange capacity is going to, that's ionic binding. And the ionic binding is much stronger and harder to break. So what they're looking at is how easy can we break those ionic bonds and make those nutrients. The macronutrients is what you're looking at more than the micronutrients. So the macronutrients of nitrogen, phosphorus, and sulfur.
Fantastic. Well, thanks a lot, Kris. Is there any final words you want to add or say?
I guess again, it's one of those things we went over a lot of details here. I have shared and we'll work with David to get it shared, not just this presentation, but a number of different documents that go over explanations and interpretation of soil test result data. And my biggest thing, and I hope that you all have gotten that with this discussion, so with this presentation and a few questions we were able to get to, is the whole idea of looking at things as a whole and understanding the complexities and interactions that are happening instead of looking at just individual numbers. So I appreciate your time this evening, and look forward to talking with all of you again sometime soon.